We will see a very important equipment for a lab that deals with biology related applications. So, this is a microfabrication course, but in the microfabrication technology, using microfabrication technology, we will be making different types of sensors, and these sensors will have practical applications as we have time and again discussed. One of the major applications that this lab focuses on, as it would be clear from the title slide of this video, is that this is a biomedical and electronic engineering systems lab. So, we tried to work at the interface of electronics and biology or engineering and biology. So, we make sensors using microfabrication technology and use those sensors to test with biological samples like tissues, cell clumps, cultured cells, cells from cell lines, etc. So, for such a scenario, it is very important that you have the facility to culture cells. What is culturing cells? It means you start off with a very small sample number of cells that you obtain from either a patient or an animal or borrowed from some other cell bank or tissue bank. These cells, then you have to provide them medium for growing, growth factors using serum, fetal bovine serum, it is called FBS, and you add these chemicals basically to these cells and give them conditions for them to grow. Once you give them the conditions to grow, they will start multiplying and dividing through their normal cell cycle. Once they divide, they form sufficient numbers and start attaching to dishes like petri dishes. So, this is the overall idea. Now, how do we grow cells? When we are growing cells, it is very important that we avoid this cell growth process or cell culturing process, to be technically correct. Cell culturing process or tissue culturing process, which also you can tell. See, cell culturing process is a very critical process, and it is very prone to contamination. What is contamination? Foreign bodies that are not supposed to be there getting into the dish where you culture cells is called contamination. So, for you to avoid such things, you need a special facility or an equipment to grow the cells. This is where the equipment that we are going to discuss comes into picture. Today we are going to see what is called a incubator, or usually it is called a CO2 incubator. So this is the CO2 incubator, you can see, correct. So, for a CO2 incubator, for cells to grow in an incubated environment, the major parameters that are required is that you should provide 37 degrees Celsius ambient temperature, 5% carbon dioxide level and 95% relative humidity. So, this equipment will make sure that these parameters are provided to the cell and they can grow without any contamination. So, first let us see what is the control panel. So, this incubator is now running. So, we have kept it on the floor. So, we have set the temperature to 37 degrees Celsius. We have set the temperature to 37 degrees Celsius, but because we have turned it on only short while ago, the control system inside it is still ramping up to the temperature. But we can always see what is the temperature to which this is set by. See here, you see buttons. These are all touch panel buttons. This is degree Celsius. So, what is the temperature that you have set? This is percentage CO2. What is the percentage CO2 that you have set? And auto start. How do you want to set it auto? The 90 degrees Celsius. What is this 90 degrees Celsius? This is for routine. As I said, contamination is a very important thing. So, we have to routinely sterilize our incubator inside. So, this 90 degrees Celsius, if you click, what it will do is, it will create 90 degrees Celsius environment inside the incubator and kill any bacteria that might be present. So, this is used for cleaning the incubator. Right now, we will not use it. Cal is for calibrating everything properly, so that if you set for 5% CO2, actually 5% CO2 should be maintained inside. So, that calibration it will set. So, let us press the degree Celsius button. So, if you press it, you will see that it is set to 37 degrees Celsius. See, it is very clearly visible. If you release it, it will show the current temperature that it is about to reach. It is slowly ramping up to 37 degrees Celsius. Likewise, if you press percentage CO2, you will see that it is set to 5% CO2. If you release it, right now you will see that it is around 5.2% CO2 is there, which is roughly equal to what we have set. Now, 95% relative humidity is maintained through another mechanism inside the incubator, which we will see shortly. So, these are the major controls in the incubator. So, these up and down arrows are used for setting these temperatures. Let us say I press this and you can if you press this, you will see the temperature, right? 37 degrees Celsius is what it's set for. If you press up down, you can actually change these temperatures like this. So you can change the values. So this is how the different values are set, and the temperatures, everything are set like this. Now let us see how the carbon dioxide is coming to the incubator. For that we have a CO2 incubator, which is, now let us see what it is. So that tank CO2, we have a cylinder, CO2 cylinder that is kept at the edge. So you can see the cylinder. So we see the cylinder which has a pressure valve that is kept at the edge. So let us see that now. So there is a pressure valve at the top. There is a tube coming out of the cylinder. That tube effectively comes and reaches the incubator. So you can see how much CO2 is left in the cylinder through gauges and valves that are there in the cylinder, and there are safety mechanisms to open and close the cylinder to make sure that there are no unnecessary explosions or any issues. So from that outlet valve, the tube will come and it will enter the incubator. Now let us see how the inside of the incubator looks like. We already have few dishes with cells kept inside the incubator. Let us see how they look like. So I am opening the incubator door now. So you see, this is how the inside looks like. We cannot keep it open for that much time, so we will quickly see the inside of it. So I am opening another door. There is a first safety door there. So this is what. Then these are compartments that you have. You have multiple 4 compartments. See, at the bottom compartment you see liquid, which is water. So we have water here. See, so this water, we have to always fill water here up to this mark where the water is there. See, there is water. We have to fill up to here. This is the water that will maintain 95% relative humidity. So always make sure that we fill it with water up to this level. Now we have cells dishes here which are being grown. So this is a petri dish, 35 mm petri dish with cells that are growing inside. It is pink in color because it is put in media and growth factors like serum. So this is Dubois minimum essential medium, DMEM with FBS, fetal bovine serum added to it, and cells are going in it. We have seen these cells in our microscope lectures how they look like. So there are different dishes which we have kept. There is another dish that we have kept which is 100 mm big one, and there is one more dish. So we are growing cells in this. So we should not keep it open for so long, because the control system will change the temperature and other parameters. So we have closed it. We have closed this window also. As it is visible, there are 3 compartments to this. So you can put lot of dishes inside, and all these dishes will consume CO2. So accordingly it will consume from the cylinder. Lesser the number of dishes, lesser the resources it will consume. It is just like us growing only. So when the cells grow, they will consume contents just like we grow. So more the number of dishes that will be inside the incubator, more will be the CO2 that will be consumed. So this is a basic working of a CO2 incubator. So as I told before, 3 main parameters: 37 degrees Celsius ambient temperature inside, 5% carbon dioxide and 95% relative humidity. Carbon dioxide is maintained through the carbon dioxide cylinder. 37 degrees Celsius is maintained through thermostats that are inside the incubator, which will maintain the temperature, and the 95% relative humidity is maintained through the water, and by maintaining it at that level which I showed you inside. And the 90 degrees Celsius option is for routinely cleansing the incubator so that contaminations are eliminated. So there is no scope for even less contamination. Contaminations has to be eliminated. There should not be no contamination as much as possible. So this is how the incubator works. It is a very important equipment in a lab that deals with biology or cultured cells, and so hope you understood how the incubator looks like and what is its functioning and how cells are kept inside. So this is something that you need to know, because you need to know where your sensors that you fabricate are applied. So if you are looking at some other field like transportation, like security, gas sensing and all, different types of equipment will be used. This is for a biology application. For that, a CO2 incubator will be used to grow the cells. Hope this was useful to you. Thank you.